Hi, my name is Hyuna Kim, and my research focuses on estimating the Antarctic ice sheet mass imbalance using data 18 of seawater from around Antarctica as a proxy. Where the ice sheet melts, it dumps both fresh water and strongly depleted data 18 into the surrounding ocean waters. By using salinity and data 18 seawater measurements, we can, in principle, differentiate between ice sheets and sea ice flux contributions. In my lab, I am using a laser cavity ring-down spectrometer to make accurate measurements of water isotopes. While the method works extremely well for freshwater samples, it is still challenging to conduct high-quality measurements for seawater due to salt contamination in the vaporizing processes. I am now developing a new method for salt water, which is based on membrane filter and water vapor standard source units. In the end, my method will be significantly faster and even portable on icebreakers as compared to the standard isotope ratio mass spectrometry method used previously. In addition, we use our instrument on rainfall samples to better understand moisture transport processes across Eastern Asia using water isotopes. Our monthly measurements are submitted regularly to the GENI project, which is a part of International Atomic Energy Administration. This data will also help us to ground truth the interpretation of our Korea speleothem samples in terms of data of calcites and water isotopes of fluid inclusions. As a part of our official GENI station monitoring requirements, we have been operating an integrated weather station since 2019. It has sensors to measure basic atmospheric parameters such as wind speed, wind direction, and air pressure, relative humidity, air temperature, and precipitation, and so on. We match those data with our GENI rainwater samples that we collect at the very same location. Especially its high temporal resolution of 10 minutes is very useful for typhoon studies.
Hello, my name is Dr. Jesper Wassenberg. I joined the IBS Center for Climate Physics last March as a Young Scientist Fellow, and I'm now in the process of launching a new speleothem research team. Our research team will focus on reconstructing past East Asian monsoon changes by analyzing the isotopic composition of water contained within speleothem carbonate called fluid inclusions. Fluid inclusion water comes from the rainfall that fell on top of the cave. Therefore, fluid inclusion isotopes such as delta 18O and delta D are a direct proxy for the isotope composition of fossil rainfall. This is a major advantage over carbonate delta 18O, which is also temperature dependent. The technique also provides a second hydrological proxy, the deuterium excess. In my new lab, I'm building a preparation line to extract a minimum of 0.1 microliter of water from speleothem samples and analyze it with a Picaro water isotope analyzer. We crush small speleothem chips at 120 degrees Celsius such that the fluid inclusions evaporate and are measured by the Picaro. These fossil rainfall isotope reconstructions can be used as a benchmark for isotope-enabled climate models. Additionally, and in combination with results from the isotope ratio mass spectrometer, we will be able to assess past cave air temperatures and seasonality of rainfall.
Welcome to the ICCP lab tour. My name is Nitesh Sinha. I am a postdoctoral research fellow here at ICCP. One of the main goals of my research is to obtain isotope-based climate reconstruction using South Korean speleothems. So I will be introducing you with our sample preparation room and mass spectrometry lab. In our sample preparation room, we cut our speleothem with ultra-precise diamond wire saw. To obtain the smoother cut, we run the saw at very low speed, and cutting a sample of about 16 to 20 centimeter takes around 2 days. Even though the wire saw cutting the surface already very clean, we prefer to further hand polish the surface to see growth in structures, impurities, identify possibly which area is containing the fluid inclusions. Once the stalagmite is cut and polished, we need to drill calcite samples for further isotopic analysis. So we typically use Dremel hand drill device to obtain samples for uranium dating, and for high precision stable isotope and clumped isotope analysis, we use our new Micromill, which can drill samples every 100 micrometer or even less. Now let me introduce you to the main equipment in our lab, our isotope ratio mass spectrometer. We have a MAC 253 Plus and Keel device from Thermo Fisher. In the Keel device, the calcite samples are transformed by acid ingestion and cleaning processes into pure CO2 gas. In this process, the isotopic composition of the calcite powder is transformed over to the CO2 gas, and then the clean CO2 gas is then injected connected through a capillary for isotopic measurement into Med253+. It measures, among many other things, carbon-13, carbon-12, and oxygen-18, oxygen-17, and oxygen-16 in ionized CO2 gas. Our system is the only instrument in Korea that can measure also the so-called clumped isotope. It's like a clumping of a heavy isotope, for instance 13C, 18O, and 16O. And the clumping is dependent on the temperature, so the clumped isotopic composition of calcite sample is a direct temperature proxy. In a sense, this instrument is a large paleothermometer. By analyzing delta 18O of calcite, we can also learn something about atmospheric circulation and the hydroclimate changes in the past. And with delta 13C of the sample, that provides insight into the soil conditions and possibly also about the vegetation's changes in the past. Our preliminary research on a stalagmite from Jungmal Cave in South Korea provides fascinating insights into a generation of an abrupt climate change event which happened around 16,000 years ago.
Welcome to our small ICCP metagenomics lab. My name is Valentina Vanghi, and the goal of my research is to link cave environments with different biological taxa that grow inside or outside the cave, such as bacteria, fungi, or plants. Eventually, we would like to expand this research to extract paleo-DNA from stalagmites and link past climate with biological changes. In my lab, I process soil and sediment samples and swabs that I collect in South Korean caves. In the first step, I determine the amount of DNA in the samples using a qubit 4 fluorimeter. Then I proceed with the library preparation, which consists of a series of chemical treatment of the DNA solution. The prepared samples are then inserted into a MinION sequencing device. The MinION is a relatively new sequencing technology, which is extremely portable and versatile. The initial DNA soup from the sample is drawn through an electrically charged membrane, which consists of thousands of nanoscale pores which bind to the DNA fragments. DNA strands are then sucked through the electrically charged nanopores. Every DNA letter, C, G, T, A, creates a slightly different electric currents, which is measured and transferred through a USB port to the computer. Eventually, the sequence of electric signals can be translated back to our DNA code. The resulting data can then be used for taxonomical identification, quantification, and other bioinformatics analysis.
After benchmarking community Earth system model simulations on various supercomputers worldwide, our ICCP supercomputer team developed a plan to launch a new climate supercomputing center in South Korea. The National Facilities and Equipment Committee of South Korea approved the funds to ICCP in 2017, and in collaboration with the IBS headquarters, we purchased in 2018 a Cray XC50 system with liquid cooling, a large parallel file system which can hold up to 9 petabytes of data, and a tape archive system with about 43 petabyte capacity. Our supercomputer, which is called Aleph, and which is located at the IBS headquarters in Daejeon, is one of the fastest academic computers in Korea. 85% of Aleph is allocated for our center, and we use Aleph extensively for a wide range of new simulations, including some of the highest resolution global warming simulations, a 100-member large ensemble simulation, a transient Pleistocene simulation, and many other numerical experiments. Together with members from the IBS headquarters, we are now discussing plans for a new supercomputing system to be purchased in 2024 with an ICCP climate modeling allocation of about 6 to 8 petaflops, factor 5 to 6 faster than our current system.